Tonight's lecture is over Chapter 10, Evolution and Natural Selection. And our learning objectives are to be able to describe examples of evolution in action. We will be able to explain Darwin's process in developing a theory of evolution. We will describe the four mechanisms that can give rise to evolution. We will also be able to discuss how populations of organisms can adapt to their environment through natural selection, and we will be able to describe the evidence for evolution. So what's the longest that you've gone without food? 24 hours? 36 hours? If you're ever hungry for long, you probably felt terrible, but humans can survive days, even weeks without food. In 1981, for example, 27-year-old Bobby Sands went on a hunger strike, forsaking all food and consuming only water. He died after 66 days without food. A fruit fly, however, can last only 20 hours or so without food. The tiny fruit fly's body doesn't hold very large caloric reserves, but could you breed fruit flies that could live longer than 20 hours on average? Yes. So, first, start with a population of 5,000 ordinary fruit flies. In biology, the term population means a group of organisms of the same species living in the same geographic region. The region can be a small area, such as a test tube, or a large area, such as a lake. In your experiment, your population occupies a plexiglass cage in your laboratory. First, remove the food from the cage. Wait until 80% of the flies have starved to death, then put a container of food into the cage. After the surviving flies eat, they'll have the energy to reproduce. When they do, collect and transfer the eggs to a new cage. So when the eggs hatch, the flies will show increased resistance to starvation. The average fly in the new generation can live for about 23 hours without food. And again, some of the flies can survive for more than 23 hours, and some for less. If you kept repeating these three steps, the average starvation resistance time would increase. After 60 generations of allowing only the flies that are best at surviving without food to reproduce, how has the population changed? Amazingly, the average fly in the resulting population can survive for more than 160 hours without food. Even the fly with the lowest starvation resistance is more than 7 times better at resisting starvation than the best fly in the original population. At the point where the food is removed, the flies in generation 60 are noticeably fatter than the flies in generation 0. What happened? In a word, evolution. That is, there was a genetic change in the population of fruit flies living in the cage. Later in this chapter, we'll fully discuss the mechanisms by which evolution can occur. In short, it is the consequence of certain individual organisms in a population being born with characteristics that make them more likely to survive and reproduce than other individuals in the same population. In the fruit fly experiment, the 20% of the flies were the most resistant to starvation could survive and reproduce, and the less starvation-resistant flies could not. Recall from our discussion of scientific thinking back in Chapter 1 that in order to give us confidence in our hypotheses about anything, those hypotheses must be testable and reproducible. Researchers carried out the 60 generations long starvation resistance experiment in fruit flies 5 separate times. In every replication, the results were the same. The fruit fly experiment answers what is sometimes perceived as a complex or controversial question: does evolution occur? The answer is an unambiguous yes. Now, suppose that we used dogs in our experiment and we only allowed the smallest dogs to reproduce. Would the average body size of individual dogs in subsequent generations decrease over time? Yes. What if the experiment were done with rabbits in a natural habitat, in the absence of human intervention, and only the fastest rabbits escape death from predation by foxes. Would the average running speed in rabbits increase over time? Yes. Each of those experiments, whether in the lab or in nature, has already been done, and in each one, evolution has been observed. And that brings us to our take-home message for section 10.1. The characteristics of the individuals present in a population can change over time. We can observe such change in nature and can cause it to occur. Charles Darwin grew up in an orderly world. In the Victorian British society he inhabited, many beliefs about humans and their place in the world had changed little over the previous 2 centuries. The Bible sufficiently explained most natural phenomena. Most people thought that the earth was about 6,000 years old and was largely unchanging. People recognized that organisms existed in groups called species or kinds. However, most thought that all species included humans, had been created simultaneously and never changed and never died out. Darwin threw into question some dearly held long-time beliefs about the natural world, and he forever changed our perspective on the origins of humans and their relationships to all other species. He didn't smash society's worldview to pieces all at once, though, and he didn't do it by himself. In the 1700s and the 1800s, scientific thought was advancing rapidly. Darwin was influenced by the work of other scientists of this era. For example, in 1778, the respected French naturalist Georges-Louis Leclerc Comte de Buffon calculated that the Earth must be about 75,000 years old, the minimum time required for the planet to cool from a molten state. Then, in the 1790s, Georges Cuvier discovered fossil remains at the bottoms of a coal slate mine that were unlike any living species. Many people, believing that biblical accounts did not allow for species to be wiped out, found Cuvier's discoveries unthinkable. His publications documented giant fossils, including the Irish elk, the mastodon, and the giant ground sloth, that bore no resemblance to any currently living animals. Cuvier was not a proponent of the idea of evolution, but the fossils he discovered allowed only one explanation. Extinction was a fact. Not only was it starting to seem that species could disappear from the face of the earth, but several scientists, including Darwin's grandfather, began to suggest that living species might change over time. In the early 1800s, the biologist Jean-Baptiste Lamarck championed a popular idea that change in living species came about chiefly through organisms' use or disuse of particular features. Think of a giraffe's neck elongating because of the need to eat leaves high in the trees. The idea was wrong, but the increased willingness among scientists to question previously sacred quote-unquote truths contributed to an atmosphere of unfettered scientific thought in which it was possible to challenge convention. Perhaps the revolutionary ideas that most inspired Charles Darwin were those of the geologist Charles Lyell. In his 1830 book, Principles of Geology, Lyell argued that geological forces had shaped the earth and were continuing to do so, producing mountains and valleys, cliffs and canyons, through gradual but relentless change. This idea that the physical features of the earth were constantly changing would most closely parallel Darwin's idea that the living species of the earth, too, were gradually but constantly changing. And that brings us to our take-home message for section 10.2. Gradual changes in scientific beliefs in the 18th and 19th centuries helped shape Charles Darwin's thinking. So, Charles Darwin was born into a wealthy family in England in 1809, and never at the top of his class, he professed to hate schoolwork. Nonetheless, at 16, he went to the University of Edinburgh to study medicine, following in his father's footsteps. Darwin was bored, though, and left at the end of his second year when the prospect of watching gruesome surgeries in the days before anesthesia was more than he could bear. Darwin then pursued the ministry studying theology at the University of Cambridge. Although he never felt great inspiration in his theology studies, Darwin was in heaven at Cambridge, where he could pursue his real love, the study of nature. Shortly after graduation in 1831, Darwin landed his dream job in the position of quote-unquote gentleman companion to the captain of the HMS Beagle on a 5-year round-the-world surveying expedition. Once on the Beagle, though, Darwin discovered that he didn't actually like sea travel. He was seasick for much of the time while he was on board, writing to his cousin, "I hate every wave of the ocean with the fervor which you can never understand." To avoid nausea, he spent as much time as possible on shore. It was there that he found field work to be his true calling. At each stop, Darwin investigated new worlds. In Brazil, he was enthralled by the tropical forests. In Patagonia, he explored beaches and cliffs, finding spectacular fossils from huge extinct mammals. Elsewhere, he explored coral reefs and barnacles, always packing up specimens for museums and recording his observations for later use. He was like a schoolboy on permanent summer vacation. Lyell's Principles of Geology was the only book Darwin took with him on the Beagle. He read it again and again, intrigued by the book's premise that the Earth is constantly changing. This idea would serve Darwin well when, a few years into its journey, the Beagle stopped at the Galapagos Islands off the northwest coast of South America. This group of volcanic islands was home to many unusual species, from giant tortoises to extremely docile lizards, which made so little effort to run away that Darwin had to avoid stepping on them. Darwin was particularly intrigued by the wide variety of birds, especially the finches, which seemed dramatically more variable than those he had seen in other locations. So, Darwin collected and donated finch specimens to the Zoological Society of London. Initially, Darwin thought that all the finches were of the same species but with different physical characteristics or traits, such as body size, beak shape, or feather color. Back in London, however, ornithologists examining Darwin's specimens could see from the birds' physical differences that the birds were not reproductively compatible. There were unique lineages for every one of the Galapagos Islands that Darwin had visited. Moreover, although the birds were of 14 different species, they all resembled very closely the single species of finch living on the nearest mainland in Ecuador. The finches were part of two important and unexpected patterns Darwin noticed on his voyage that would be central to his discovery of a mechanism for evolution. The first pattern, that the different island finches all resembled the mainland species, seemed a suspicious coincidence to Darwin. Perhaps they used to be a part of the same mainland population. Over time, they may have separated and diverged from the original population and gradually formed new but similar species. Darwin's logic was reasonable, but his idea flew in the face of all the scientific thinking of the day. The second pattern Darwin noticed was that throughout his voyage, there was a striking similarity between the fossils of extinct species and the living species in that same area. In Argentina, for instance, he found giant fossils from a group of organisms called glyptodonts. The extinct glyptodonts, though huge, resembled armadillos, a species that still flourished in the same area. If glyptodonts had lived in South America in the past and armadillos were currently living there, why was only one species living? And why were the glyptodont fossils found only in the places where modern armadillos lived? Darwin deduced that glyptodonts resembled armadillos because they were their ancient relatives. It was a logical deduction, but contradicted the scientific dogma of the day that species were unchanging and extinction did not occur. So, Darwin had no eureka moment about evolution while he was on the Beagle, but after his voyage, inspiration struck when he was reading for amusement essay on the principle of population by economist Thomas Malthus. Malthus calculated that populations could grow much faster than food supplies with disastrous consequences. Darwin speculated that rather than all sharing the same doom, maybe the best individuals would quote-unquote win the struggle for existence and the worst would quote-unquote lose. If so, he suddenly saw favorable variations would tend to be preserved and unfavorable ones would be destroyed. In 1842, Darwin prepared a draft of his ideas in a 35-page paper, and he fleshed it out over the next couple of years. He had an inkling that his idea would rock the world. In a letter to a close friend, he wrote, "At least the gleams of light have come, and I almost convinced, quite contrary to the opinion I started with, that species are not. It is like confessing to murder, immutable." Inexplicably, he put his manuscript into a drawer where it remained for 14 years, even as his friends warned him that he should probably publish his ideas in case someone else came up with the same ideas. Then in 1858, Darwin found that his friends' warnings had come true with a vengeance. So in a letter to Darwin, Alfred Russel Wallace, a young British biologist in Malaysia, laid out a clear description of the process of evolution by natural selection after having read Malthus's book. Because they had corresponded previously, Wallace asked Darwin to, quote, publish it if you think it is worthy, unquote. Crush it having been scooped, Darwin wrote that all of my originality will be smashed. Darwin's scientist friends, however, arranged for a joint presentation of Wallace's and Darwin's work to the Linnaean Society of London. As a result, both Darwin and Wallace are credited for the description of evolution by natural selection. Darwin then rapidly completed a full book. In 1859, he published On the Origin of Species by Means of Natural Selection, or The Preservation of Favored Races in the Struggle for Life. The book was a hit, selling out on its first day, provoking public discussion and debate, and ultimately causing a wholesale change in the understanding of many important evolutionary ideas. Where once the worldview was of a young Earth, populated by unchanging species, all created at one time, with no additions or extinctions, now there was a new dynamic view of life on Earth, descent with modification. Species could and did change over time, and as some species split into new species, others became extinct. Darwin's theory has proved to be among the most important and enduring contributions in all of science. It has stimulated an unprecedented diversity of theoretical and applied research, and it has withstood repeated experimental and observational thinking. And that brings us to our take-home message for section 10.3. On a voyage around the world, Darwin noted unexpected patterns among fossils and living organisms. Fossils resembled but were not identical to living organisms in the same area, and finch species on each of the Galapagos Islands differed from one another in significant ways. These observations helped Darwin develop his theory of how species might change over time. So, suppose you are put in charge of a large population of tigers in a zoo. Almost all of them are orange and brown with black stripes. Occasionally, though, unusual all-white tigers are born. The white phenotype is the result of the presence of a rare pair of alleles that suppresses the tiger's production of most fur pigment. So remember from a previous chapter that alleles are variants of a gene, and that most mammals inherit one allele from their mother and one allele from their father for each gene. Because visitors flock to zoos to see white tigers, you want to increase the proportion of your tiger population that is white. How would you do so? To produce more animals with the white phenotype, you could breed the white tigers with each other, and as the generations go by, your population will include a higher proportion of white tigers. When this happens, you will have witnessed evolution, a change in the proportion of alleles for the pigment suppression gene in the population. Alternatively, you could increase the proportion of white tigers in your population by acquiring some white tigers from another zoo. Through this simple addition of white tigers to your population, the population will include a higher proportion of white tigers. When you add white tigers or remove orange tigers, you will again witness evolution in your population. As this example illustrates, evolution does not involve changing the genetics or physical features of individuals. Individuals do not evolve. Rather, evolution happens when the proportions of the alleles in the population change, and alleles frequency is the proportion in which it is present in a population relative to the other alleles of the same gene. Think of this as the alleles market share. In the tiger example, the white fur alleles initially had little market share, perhaps as small as 1%. Over time, though, the white fur alleles came to make up a larger and larger proportion of total fur pigment alleles in the population, and as this happened, evolution occurred. So, any time an allele's market share changes, evolution is taking place. Perhaps the most dramatic way we see this change is in acts of predation. When one organism kills another, the dead animal's alleles will no longer be passed on in the population, and if certain alleles alter coloration or speed or some other defense mechanism that makes an animal more likely to be killed by another, those alleles are likely to lose market share. The converse is equally true. Evolution occurs when alleles increase in frequency. Keeping in mind that evolution is genetic change in a population, let's explore the 4 processes that can lead to such genetic changes. So remember, I want to say it clearly. Evolution only occurs on the scale of the population. And that brings us to our take-home message for section 10.4. Evolution is a change in allele frequencies within a population. It often occurs by 4 different mechanisms: mutation, genetic drift, migration, and natural selection. So, mutation is an alteration of the base pair sequence of an individual's DNA. When this alteration occurs in DNA that is part of a particular gene, the change in the DNA sequence may cause a change in an allele that has important consequences. Only mutations that affect reproductive cells can be passed from parent to offspring and have the potential to alter the allele frequencies within a population. As an example, suppose that a mutation changes one of a person's 2 blue eye alleles into a brown eye allele. If this mutation occurs in the sperm or egg-producing cells that can be passed on to the next generation, the offspring may carry the brown allele. When this happens, the proportion of the blue eye alleles in the population is slightly reduced, and the proportion of brown eye alleles is slightly increased. Thus, evolution has occurred. Mutations can occur spontaneously during cell division, or as we saw in Chapter 6, they can be induced by a variety of environmental factors, including radiation such as X-rays and some chemicals. But although these factors can induce mutations and influence the rate at which mutations occur, they do not generally influence exactly which mutations occur. Thus, we tend to say that mutations are random. So, in other words, 1, we cannot predict which individuals will have which mutations, and 2, we cannot predict whether the consequence of a mutation will be neutral with no effect, harmful, or useful. Treatment of an agricultural pest with certain chemicals, for example, might increase the number of mutations in that pest population, but it does not increase the likelihood that a particular mutation is beneficial or detrimental. This does not mean, however, that all mutations have an equal probability of occurring. Some mutations are far more likely to occur than others. So, mutation plays a critically important role relevant to all mechanisms of evolution, including natural selection. Mutation is the ultimate source of genetic variation in a population. In the previous blue eye and brown eye example, we saw that a mutation may lead to the conversion of one allele to another that is already found within the population. Even more importantly, a mutation may create a completely novel allele that codes for the production of a new protein. That is, a change in the base pair sequence of a person's DNA may cause the formation of a gene product that has never existed before in the human population. Instead of blue or brown eyes, the mutated gene might code for yellow or red eyes. If such a new allele occurs in the reproductive cells, and if it does not significantly reduce an individual's reproductive fitness, this is a topic we'll talk about later, the new allele can be passed on to the offspring and thus remain in the population. At some future time, the mutation might even confer higher fitness, in which case natural selection could lead to an increased frequency of this allele in the population. For this reason, mutation is critical to natural selection. All variation, the raw material for natural selection, must initially come from mutation. Despite this vital role in generating variation, nearly all mutations either have no impact on an organism's fitness or have an adverse impact by causing early death or reducing the organism's reproductive success. Mutation in a typically functioning allele that codes for a typically functioning protein usually result in either an allele that codes for the same protein or a new allele that codes for a non-functioning protein. The latter case almost inevitably reduces an organism's fitness. For this reason, our bodies protect our sperm or egg-producing DNA with a variety of built-in error correction mechanisms. As a result, mutations are rare in humans on the order of one mutation in every 30 million base pairs in each generation. And that brings us to our take-home message for section 10.5. Mutation is an alteration of the base pair sequence in an individual's DNA. If such an alteration changes an allele in an individual's gamete-producing cells, the frequency of alleles in the population changes, and the population evolves. Mutation is the only way that new alleles can be created within a population. So, let's consider two alleles for a particular trait such as white spotting in a cat's fur. A single gene trait with two alleles for which the white spotting allele is dominant over the non-white spotting allele. Neither allele confers higher or lower fitness than the other. Now suppose that two heterozygous parent cats have one offspring. Which combination of alleles will that kitten receive? This is impossible to predict because it depends completely on which sperm fertilizes which egg. The luck of the draw. If the parent's sole offspring inherits a recessive allele from each parent, will the population's allele frequencies change? Yes, the new individual's two recessive alleles increase the proportion of the recessive alleles in the population. It is equally likely that the parent's only offspring will receive two dominant alleles. In either case, because a change in allele frequencies has occurred, evolution has happened. This is an example of the second evolutionary mechanism, genetic drift, which is a random change in allele frequencies in a population. A change in allele frequencies due to genetic drift is not related to the allele's influence on reproductive success. This impact of genetic drift is much greater in small populations than in large populations. In a large population, any one couple having an offspring with two recessive alleles is likely to be offset by another couple having an offspring with two dominant alleles. When that happens, the overall allele frequencies in the population do not change and no evolution has occurred. One of the most important consequences of genetic drift is that it can lead to fixation for one allele of a gene in a population. Fixation results when an allele's frequency in a population reaches 100% and the frequency of all other alleles of that gene becomes 0%. If this happens, there is no more variability in the population for this gene. All individuals always produce offspring carrying only that allele until new alleles arise through mutation. Because of fixation, genetic drift reduces the genetic variation in a population. Multiple evolutionary mechanisms can occur simultaneously. An allele favored by natural selection, for example, may simultaneously increase or decrease in frequency due to genetic drift. Two special cases of genetic drift, the founder effect and population bottleneck effect, are important in the evolution of many populations. So let's first discuss the founder effect. A small number of individuals may leave a population and become the founding members of a new isolated population. This founder population may have different allele frequencies than the original source population, particularly if the founders make up a small group. If this new population does have different allele frequencies, evolution has occurred. Because the founding members of the new population will give rise to all subsequent individuals, the new population will be dominated by the genetic features that happen to be present in that group of founding mothers and fathers. This type of genetic drift is called the founder effect. The Amish population in the United States was well established by a small number of founders, some of whom happened to carry the allele for Ellis-Van Creveld syndrome. This syndrome leads to phenotypic effects, including polydactyly, the condition of having extra fingers and toes. As a consequence, this trait, while rare, now occurs much more frequently among the Amish than it does worldwide. And that brings us to the population bottleneck effect. Occasionally, a famine, natural disaster, or rapid environmental change causes the deaths of a large proportion of individuals in a population. Because the population is quickly reduced to a small fraction of its original size, this reduction is called the bottleneck effect. If the catastrophe is equally likely to strike any member of the population, the remaining members are essentially a random, small sample of the original population. For this reason, the remaining population may not possess the same allele frequencies as the original population. Thus, the consequence of such a population bottleneck would be evolution through genetic drift. Just such a population bottleneck occurred with cheetahs near the end of the last ice age about 10,000 years ago. Although the case is unknown, possibly environmental cataclysm or human hunting pressures, it appears that nearly all cheetahs died. Although the population subsequently rebounded, all cheetahs living today can trace their ancestry to a dozen or so lucky individuals that survived the bottleneck. As a result of this past instance of evolution by genetic drift, there is almost no genetic variation in the current population of cheetahs. And that brings us to our take-home message for section 10.6. Genetic drift is a random change in allele frequencies within a population unrelated to the allele's influence on reproductive success. The effects of genetic drift are more significant in small populations. And that brings us to our third mechanism of evolution. This one is called migration, and it can also be called gene flow. And this is the movement of some individuals of a species from one population to another. This movement distinguishes migration from the founder effect, in which individuals migrate to a new habitat previously unpopulated by that species. If migrating individuals survive and reproduce in the new population, and if they carry a different proportion of alleles than the individuals in their new home, then the recipient population experiences a change in allele frequencies, and consequently, this population experiences evolution. And because alleles are simultaneously lost from the population that the migrants left behind, that population too experiences a change in its allele frequencies and thus evolves. Gene flow between two populations is influenced by the mobility of the organisms and by environmental barriers such as mountains or rivers. And as we'll see in an upcoming chapter, human activities can dramatically influence the migratory potential of organisms as we transport species from one place to another. This can happen intentionally, such as when people purchase exotic animals to keep as pets or bring in non-native species for their gardens, or unintentionally, such as when marine species get trapped in the ballast tanks of cargo ships, later to be released in the ports on the other side of the world when the tanks are flushed. And that brings us to our take-home message for section 10.7. Migration leads to a change in allele frequencies in a population as individuals move into or out of the population. And that brings us to our final mechanism, which is natural selection. Natural selection is the mechanism that Darwin identified in The Origin of Species, in which he noted that three conditions are necessary for natural selection to occur. And they are variation for a trait, heritability, and differential reproductive success. So let's talk about condition 1, variation for a trait. Observing people with their dogs at a dog park, you will see dogs of all shapes, colors, and sizes. The humans, too, will vary in facial features and a multitude of other characteristics. Variation is all around us. Beyond making the world an interesting place to live in, variation serves another purpose. It is the raw material on which evolution works. Variation is not limited to physical features such as fur color or face shape. Individuals vary in physiological and biochemical ways too. Some people can quickly and efficiently metabolize alcohol, for example, while others become violently ill soon after sipping a glass of wine. Similarly, we vary in our susceptibility to poison ivy or to diseases such as malaria. Behavioral variation, from temperament to learning abilities to interpersonal skills, is dramatic and widespread as well. So impressed was Darwin with the variation he observed throughout the world that he devoted his first 2 chapters of The Origin of Species to a discussion of variation in nature and among domesticated animals. And that brings us to condition 2, heritability. For natural selection to act on a trait, offspring must inherit the trait from their parents. Although inheritance was poorly understood in Darwin's time, it was not hard to see that for many traits, offspring looked more like their parents than like some other random individual in the population. Animal breeders had long known that the fastest horses generally give birth to the fastest horses. Farmers understood that plants with the highest productivity generally produce seeds from which highly productive plants grow. We call this transmission of traits from parents to offspring through genetic information inheritance or heritability. And that brings us to our third condition, which is differential reproductive success. Darwin derived that third condition for natural selection from 3 fairly simple observations. First, more organisms are born than can survive. Second, organisms are continually struggling for existence. Third, some organisms are more likely than others to survive and reproduce. In a world of limited resources, finding food or shelter is a zero-sum game. If one organism is feasting, another is likely to be starving. For example, in the food experiments with the fruit flies, we saw that the flies inheriting the ability to pack on fat when food was available had greater reproductive success than those inheriting a poor ability to store fat. So that's it. Natural selection, one of the most influential and far-reaching ideas in the history of science, takes place when these three basic conditions are met. Over time, the traits that lead some organisms to have greater reproductive success than others will increase in frequency in a population, while traits that reduce reproductive success will become less and less common. So, some traits specifically confer greater reproductive success because they make the individual more attractive to the opposite sex. This is called sexual selection. Such traits, including brightly colored feathers of male peacocks, the large antlers of male red deer, and a variety of other ornaments that increase in individual status or appeal, rise in frequency because they satisfy the three conditions for natural selection. So another way of looking at it is that natural selection is to focus not on the winners who are producing the most offspring, but on the losers. Natural selection can be viewed as the elimination of some heritable traits from a population. If you carry a trait that makes you a slower running rabbit, for example, you are more likely to be eaten by a fox. If that running speed is a heritable trait, and it is, the next generation in a population contains fewer slow rabbits. Over time, the population is changed by natural selection. In other words, it evolves. One of Darwin's contemporaries, Thomas Huxley, supposedly said after reading The Origin of Species that he couldn't believe he didn't figure it out himself. Each of the three basic conditions is indeed simple and obvious. The brilliant deduction, though, was to put the three together and appreciate the consequences. And that brings us to our take-home message for section 10.8. Natural selection is a mechanism of evolution that occurs when there is heritable variation for a trait, and individuals with one version of the trait have greater reproductive success than do individuals with a different version of the trait. So, do recessive traits gradually become less common in a population? In the early 1900s, there was much discussion among biologists about this question. They wondered, if the allele for brown eyes is dominant over the allele for blue eyes, why doesn't a population eventually become all brown-eyed? Well, R.C. Punnett, the originator of the Punnett square that we described in Chapter 9, posed this question to G.H. Hardy, a mathematician with whom Punnett played cricket. Hardy replied the next day, and he published his answer in Science in 1908. It turns out that Wilhelm Weinberg had answered the question independently 6 months earlier, but because he published his findings in an obscure journal, scientists didn't appreciate his result for another 35 years. Their result underlies all evolutionary genetics and is referred to as the Hardy-Weinberg law. It reveals several things, including that the answer to the question of whether recessive traits become rarer in a population is no. A trait does not decrease in frequency simply because it is recessive. Here's how Hardy and Weinberg demonstrated it. First, we refer to the frequency of the dominant allele in the population as P and the frequency of the recessive allele as Q. We're assuming that the gene has only two alternative alleles in the population. The equations can be expanded for genes with more alleles without changing the overall outcomes. Since every allele in the population has to be either dominant or recessive, we can say that P plus Q equals 1. The percentage of the two alleles must total 100%. If we know the frequency of either allele in the population, we can subtract it from 1 to calculate the frequency of the other. So, next, we can predict how common each genotype in the population will be. The frequency of homozygous dominant is just the probability that an individual gets two copies of the dominant allele, which is P times P, or P squared. Applying the same math, the frequency of homozygous recessive individuals in the population is simply Q times Q, or Q squared. Predicting the frequency of heterozygous individuals is slightly more complicated. Because the dominant allele may come from either the mother or the father, the frequency of the heterozygous genotype is 2 times P times Q, or 2PQ. In other words, it's the sum of the frequency of the heterozygous genotype. For example, getting the dominant allele from the mother and the frequency of the heterozygous genotype, for example, getting the dominant allele from the father, which is P times Q plus Q times P, which simplifies to 2PQ. So, consider an example. Suppose a population of 1,000 kangaroo rats has the following phenotype and genotype frequencies. 16 are dark brown, which they represent the homozygous dominant allele pair. 222 are spotted. They represent the heterozygous portion of the population, and 762 are light brown, and that 762, the light brown ones, represent the homozygous recessive portion of the population. The trait shows incomplete dominance, with the allele for dark brown color dominant over the allele for light brown color, and the heterozygote having a spotted phenotype. Because every individual in the population has two alleles for the coat color gene, one from each parent, there are twice as many alleles as members of the population. So a population of 1,000 kangaroo rats has 2,000 alleles of the gene for coat color. Now let's look at the allele frequencies. Each homozygous dominant individual has two copies of the dominant allele, and each heterozygous individual has one copy. If that is the case, the frequency of the dominant allele can be calculated as 2 times 16 plus 222 divided by 2,000, which equals 0.127, and that equals P. And we can calculate the frequency of the recessive allele the same way. 2 times 762 plus 222 divided by 2,000 equals 0.873, and that is our Q number. And be careful on these equations, because you got to make sure that you've got the brackets and the parentheses in the right place and you do your order of operations properly. Since we know the allele frequencies, P is 0.127 and Q is 0.873, we can determine the genotype frequencies of the offspring produced in this population. According to the equation given earlier, they should be a frequency of P squared equaling 0.127 squared, which equals 0.016 for the frequency of heterozygous dominant individuals in this population. Then, to calculate the frequency of heterozygous individuals in this population, 2PQ equals 2 times 0.127 times 0.873, which gives us 0.222. And the frequency of homozygous recessive individuals looks like this. It's Q squared, which equals 0.873 times 2, and that equals 0.762. So, if 1,000 kangaroo rats are produced, we would expect to see genotype frequencies that are the same as those in the parent generation, which is 16 of the homozygous dominant, 222 of the heterozygotes, and 762 of the homozygous recessive individuals. And from these genotype frequencies, we would expect the following allele frequencies among the offspring they produce. So the frequency of the dominant allele would be 2 times 16 plus 222 divided by 2,000 equals 0.127 equals P. And the frequency of the recessive allele, which is 2 times 762 plus 222 divided by 2,000 equals 0.873, or in other words, it equals Q. So notice that the frequencies are unchanged. The allele frequencies 0.127 and 0.873 will always produce the same genotype frequencies. 0.16, 0.22, and 0.762, which in turn will always have the same allele frequencies. Put another way, the recessive allele does not decrease in the population over time. Our example shows that if we know the frequency of each allele in a population, we can predict the genotypes and phenotypes we should see in that population. However, the Hardy-Weinberg equations and predictions are based on two key assumptions: that random mating occurs with alleles randomly coming together in a possible genotype, and that there is no evolution. Moving on, if individuals are dying off specifically because they carry the recessive allele or because they carry the dominant allele, the allele frequencies would change due to natural selection. Likewise, other mechanisms of evolution, mutations, migrations, or genetic drift would alter the allele frequencies. In each of these exceptions, the allele frequencies would change through evolution. What if we examine a population and observe genotype frequencies that are not those predicted by the Hardy-Weinberg equations? If this occurs, we say that the population is not in Hardy-Weinberg equilibrium, and we know that either evolution or non-random mating is occurring, or both. Our calculations can help us better understand the forces influencing the population and suggest further lines of investigation. The proportion of ingredients is important, but the final result is also a matter of how you put them together. Equilibrium is key. Alain Ducasse, French chef. And that brings us to our take-home message for section 10.9. Knowing the frequencies of each allele in a population, we can predict the genotypes and phenotypes we should see. If there is random mating and no evolution is occurring in a population, recessive and dominant alleles do not change in their frequencies. Moving on, survival of the fittest. This is perhaps one of the most famous but most misunderstood and misused phrases. For starters, quote-unquote survival of the fittest was not coined by Darwin, but by Herbert Spencer, an influential sociologist and philosopher. In fact, the phrase did not even appear in The Origin of Species when Darwin first published it. It wasn't until the 5th edition, 10 years later, that he used the phrase in describing natural selection. As we'll see, in evolution, the word fitness is not defined by an organism's ability to survive or its physical strength or its health. Rather, fitness is a measure of the amount of reproduction, the reproductive output of an individual with a particular phenotype relative to the reproductive output of individuals of the same species with alternative phenotypes. To see how this works, let's consider two fruit flies. One fly carries the genes for a version of the trait that allows it to survive a long time without food. The other has the genes for a different version of the trait that allows it to survive only a short while without food. Which fly has the greater fitness? If the environment is characterized by long periods without food, the fly that can live a long time without food is likely to produce more offspring than the other fly. So over the course of its life, it has greater fitness. The alleles carried by an individual with high fitness will increase their proportion in a population over time, and the population will evolve. An organism's fitness encompasses three important elements. First, an individual's fitness is measured relative to other genotypes or phenotypes in the population. Those traits that confer the highest fitness will generally increase in frequency in a population, and their increase will always come at the expense of alternative traits that confer low fitness. Second, fitness depends on the specific environment in which the organism lives. The fitness value of having one trait versus another depends on the environment. With a sand-colored mouse living in a beach habitat will be more fit than a chocolate-colored mouse. But the same sand-colored mouse will be vulnerable to predators if it lives in darker brush away from the beach. An organism's fitness, although genetically based, can change over time and across habitats. Thirdly, fitness depends on an organism's reproductive success compared with other organisms in the population. If you carry an allele that gives you the trait of surviving for 200 years, but also causes you to be sterile, your fitness is zero. That allele will never be passed down to future generations. On the other hand, if you inherit an allele associated with a trait that causes you to die at the age of everyone else, but also causes you to have twice as many offspring as the average individual, your fitness is increased. The phrase, quote-unquote, survival of the fittest, is misleading because it is the individuals with the greatest reproductive output that are the most fit in any population. It becomes a more meaningful phrase if we apply it to alleles rather than individual organisms. Those alleles that increase an individual's fitness will survive in a population more than those that decrease an individual's fitness. And that brings us to our take-home message for section 10.10. Fitness is a measure of the reproductive output of an individual with a particular phenotype compared with the reproductive output of individuals with alternative phenotypes. An individual's fitness can vary depending on the environment in which it lives. So, if you put a group of humans on the moon, would they flourish? If you took a shark from the ocean and put it in your swimming pool, would it survive? In both cases, the answer is no. Organisms are rarely successful when put into novel environments, and the stranger the new environment, the less likely an organism is to survive. Why? Well, as Darwin noted during his travels, the organisms that possess traits that allow them to better exploit the environment in which they live tend to produce more offspring than the organisms with alternative traits. With passing generations, a population will be made up of more of these fitter organisms, and as a consequence, populations of organisms will tend to be increasingly well matched or adapted to their environment. Adaptation refers both to the process by which organisms become better matched to their environment and to the specific features that make an organism more fit. Examples of adaptations abound. Bats have an extremely accurate type of hearing called echolocation for navigating and finding food even in complete dark. Porcupine quills make porcupines almost impervious to predation. Mosquitoes produce chemicals that prevent blood from clotting so that they can extract blood from other animals. But although natural selection can produce adaptation, it doesn't necessarily lead to organisms perfectly adapting to their environment. In Lewis Carroll's Through the Looking Glass, the Red Queen tells Alice that in this place it takes all the running you can do to keep in the same place. She might have been speaking about the process of evolution by natural selection. Even as less fit individuals are continuously weeded out of the population, natural selection doesn't result in populations of organisms perfectly adapted to their environment. That is where the Red Queen's wisdom comes in. So, consider one of the clearly documented cases of evolution in nature, the beak size of Galapagos finches. Over multiple decades, biologists Rosemary Grant and Peter Grant closely monitored the average size of the finches' beaks. They found that the average beak size within a population fluctuated according to food supply. During dry years, when the finches had to eat large, hard seeds, birds with bigger, stronger beaks were more successful and multiplied. During wet years, smaller-beaked birds were more successful because there was a surplus of small, soft seeds. The ever-changing, quote-unquote, average finch beak illustrates that adaptation does not simply march towards some optimal endpoint. Evolution in general, and natural selection specifically, does not guide organisms toward betterness or perfection. Natural selection is simply a process by which, in each generation, the alleles that cause organisms to have the traits that make them most fit in that environment at that time tend to increase in frequency. If the environment changes, the alleles that are most favored may change too. I was taught that the human brain was the crowning glory of evolution so far, but I think it's a very poor scheme for survival. Kurt Vonnegut, American writer. So, in the penultimate paragraph of The Origin of Species, Darwin wrote, "As natural selection works solely by and for the good of each being, all corporeal, that is physical, and mental endowments will tend to progress towards perfection." In this passage, he overlooks several factors that prevent populations from progressing inevitably towards perfection. 1. Environments change quickly. Natural selection may be too slow to adapt the organisms in a population to such a moving target. 2. Variation is needed as the raw material of natural selection. A mutation creating a new quote-unquote perfect version of a gene may never arise. And 3. Different alleles for a trait may each cause individuals to have the same fitness. In this case, each allele represents an equally fit solution. And that brings us to our take-home message for section 10.11. Adaptation refers both to the process by which organisms can become better matched to their environment and to the specific traits that make an organism more fit. Although natural selection can produce adaptation, it doesn't necessarily lead to organisms perfectly adapted to their environment. So, plant and animal breeders understood natural selection long before Darwin did. They just didn't know that they understood it. For instance, farmers bred crops for maximum yield, and dog, horse, and pigeon breeders selectively bred animals with their favorite traits to produce offspring with more exaggerated versions of those traits. The process used by animal breeders and farmers is called artificial selection, but the underlying genetic process does not differ from natural selection because the three conditions are satisfied. Artificial selection can be thought of as a special case of natural selection, however, because the differential reproductive success is determined by humans rather than by nature, and artificial selection, unlike natural selection, is typically goal-oriented. Apple growers, for example, use artificial selection to produce the wide variety of apples now available. It was a stroke of genius for Darwin to recognize that the same process farmers were using to develop new and better crop varieties is occurring naturally in every population on Earth, and always has been. So, certain traits are easily categorized. White versus orange tiger fur, for example. Other traits, such as height, are influenced by many genes and environmental factors, and a continuous range of phenotypes occurs. Whether influenced by one gene or by a complex interaction of many genes and the environment, a trait can be subject to natural selection and changed in any of several ways. The first way we'll discuss is directional selection. In directional selection, individuals with one extreme of the range of variation in the population have higher fitness. Hundreds of experiments in nature, in laboratories, and on farms demonstrate the power of directional selection. Turkeys, for example, have been selected for increased size of their breast muscles, which makes them more profitable for farmers. Directional selection has been so successful that birds' breast muscles are now so large that it has become impossible for them to mate. All turkeys on large-scale industrial poultry farms must now reproduce through artificial insemination. The next one is stabilizing selection. How much did you weigh at birth? 5 pounds? 10 pounds? The record birth weight was 22.5 pounds. Unlike directional selection, which leads to increased fitness at one extreme and reduced fitness at the other, stabilizing selection takes place when individuals with intermediate phenotypes are the most fit. The death rate among newborns, for example, is lowest at birth weights between 7 and 8 pounds, and is higher for both lighter and heavier babies. The variation in birth weight has decreased as stabilizing selection has reduced the frequencies of genes associated with high and low birth weights. Interestingly, modern technologies including cesarean deliveries and neonatal intensive care units allow many babies to live who would not have survived without such technology. This has the unintended consequence of reducing the selection against any alleles causing these extreme traits, thereby reducing the rate of their removal from the population. And that brings us to disruptive selection. In disruptive selection, individuals with extreme phenotypes experience the highest fitness, and those with intermediate phenotypes have the lowest fitness. Among some species of fish, coho salmon, for example, only the largest males acquire good territories. They generally enjoy relatively high reproductive success, while the intermediate-sized fish regularly get run out of the good territories. Some of the tiny males are able to sneak in and fertilize eggs before the territory owner detects their presence. Consequently, we see an increase in the frequency of small and large fish, but a reduction in the frequency of medium-sized fish. So that brings us to our take-home message for section 10.12. Animal breeders and farmers undertaking artificial selection are making use of natural selection when they modify their animals and crops through selective breeding. In directional selection, the average value for the trait increases or decreases. In stabilizing selection, the average value of the trait remains the same, while extreme versions are selected against. And in disruptive selection, individuals with extreme phenotypes have the highest fitness. Moving on, we have seen that natural selection can change allele frequencies and affect how often simple traits, such as fur color or turkey breast size or plant height, appear in a population. But what about complex traits, such as behaviors that involve numerous physiological and neurological systems? Are they also shaped by natural selection? The short answer is yes. Remember, evolution by natural selection, changing the allele frequencies for traits, is occurring whenever 1. There is a variation for the trait, 2. That variation is heritable, and 3. There is differential reproductive success based on that trait. These conditions can easily be satisfied for complex traits, including behaviors. In 1954, to address this question, William Thompson trained a group of rats to run through a maze for food. He found a huge amount of variation in the rats' abilities. Some rats learned much more quickly than others how to run the maze. Thompson then selectively bred the fast learners with each other and the slow learners with each other, over several generations, he developed two separate populations. Rats descended from a line of fast maze learners, and rats descended from a line of slow maze learners. After only 6 generations, the slow learners made twice as many errors as the fast learners before mastering the maze, while the fast learners were adept at solving complex mazes that would give many humans some difficulty. More than 60 years later, it's still unclear which particular genes are responsible for maze-running behavior, yet the selection experiment demonstrates that this behavior has a strong genetic component. So, natural selection can also produce complex traits in unexpected, roundabout ways. One vexing case involves the question of how natural selection could produce an organ as complex as a fly wing when 1% or 2% of a wing, that is, an incomplete structure, doesn't help an insect to fly. The key to answering this question is that 1% of a wing doesn't need to function as a wing to increase an individual's fitness. Often, structures are enhanced or elaborated on by natural selection because they enhance fitness by serving some other purpose. Experiments using models of insects demonstrated that a small percentage of a wing in the form of a nub or almost wing confers no benefit at all when it comes to flying. The nubs don't even help the flies keep their orientation during a controlled fall. The incipient wings do, however, help the insect to address a completely different problem. They allow much more efficient temperature control so that an insect can gain heat from the environment when the insect is cold and dissipate heat when the insect is hot. Experiments on heat control efficiency show that as small nubs become more and more pronounced, they are more effective, but only up to a point. Eventually, the thermoregulatory benefit stops increasing even if the nub length continues to increase. But right around this point, the proto-wing starts to confer some aerodynamic benefits. Consequently, natural selection may continue to increase the length of this almost wing, but now the fitness increase is due to a wholly different effect. Such functional shifts explain the evolution of numerous complex structures. In the fossil record, for example, flightless ancestors of modern birds possess feathers possibly for insulation, and flexible joints made of fibrous tissue that hold together the skull bones, allowing mammals to more easily pass through the birth canal, are also found in vertebrates that hatch, allowing for growth in these animals. And that brings us to our take-home message for section 10.14. Natural selection can change allele frequencies for genes involved in complex physiological processes and behaviors. Sometimes a trait that has been selected for a particular function is later modified to serve a completely different function. And now we'll start to discuss the evidences of evolution. There are 5 primary lines of evidence for evolution. One is the fossil record. Second is biogeography. Third is comparative anatomy and embryology. Fourth is molecular biology. And fifth is laboratory and field studies. It is indeed remarkable that this theory, evolution, has been progressively accepted by researchers following a series of discoveries in various fields of knowledge. The convergence, never sought nor fabricated, of the results of work that was conducted independently is in itself a significant argument in favor of this theory. Pope John Paul II, 1996. In the 160 or so years since Darwin first published The Origin of Species, thousands of studies have been conducted testing his theory of evolution by natural selection, both in the laboratory and in natural habitats. A wide range of modern methodologies, such as genome sequencing algorithms for comparing genome similarities and constructing the most likely phylogenies, contribute to a much deeper understanding of the process of evolution.
This ongoing accumulation of evidence overwhelmingly supports the basic premise that Darwin put forward, while filling many of the gaps that frustrated him. So let's first look at the fossil record. Although it has been central to much documentation of evolution, it is very incomplete. After all, the soft parts of an organism tend to decay rapidly and completely after death. And there are only a few environments, such as tree resin, tar pits, the bottom of deep lakes, and continental shelves, in which the processes of erosion and decomposition are so reduced that an organism's hard parts, including bones, teeth, and shells, can be preserved for thousands or even millions of years. These remains, called fossils, can be used to reconstruct what organisms looked like long ago. Such reconstructions often provide a clear record of evolutionary change, including insights into the patterns and processes of history. So, the use of radiometric dating paints a clearer picture of the organism's evolutionary history, revealing the age of the rock in which the fossil is found. In Darwin's time, it was assumed that the deeper in the earth a fossil was found, the older it was. Radiometric dating makes it possible to determine not just the relative age of fossils, but also their absolute numerical age. Dating is done by evaluating the amounts of certain radioactive isotopes present in the fossil-containing rocks or the layers above and below them. Radioactive isotopes in a rock begin breaking down into more stable compounds as soon as the rock is formed, and they do so at a constant rate. Nothing alerts this rate. We can calculate the age of the rock and thus the age of the fossil by measuring the relative amounts of the radioactive isotope and its leftover decay product in the rock where a fossil is found. Radiometric dating confirms that the Earth is very old. Rocks more than 3.8 billion years old have been found on every continent, with the oldest so far found in northwestern Canada. In addition, 4.4 billion year old minerals have been found in Australia. By measuring radioactive isotope uranium-238, which has a half-life of 4.5 billion years, researchers have determined that the Earth is about 4.6 billion years old and that the earliest organisms appeared at least 3.5 billion years ago. Radiometric dating also makes it possible to put the fossil record in chronological order. By dating all the fossils discovered in one locale, paleontologists can better evaluate whether the organisms are related to one another and how groups of organisms changed over time. So, paleontologists recognize that fossilization is an exceedingly unlikely event, as discussed earlier, and when it does occur, it represents only those organisms that 1. Happen to live in that location, 2. Could be preserved under certain chemical conditions, and 3. Had physical structures that can leave fossils. For this reason, the fossil record is unavoidably incomplete. Entire groups of organisms have left no fossil record at all, and for others, the record has numerous gaps. Despite the shortcomings, the fossil record can be incredibly detailed and interesting, and includes fossils that link every major group of vertebrates. So, the evolutionary history of horses is among the most well-preserved in the fossil record. Horses first appeared in North America about 55 million years ago and radiated around the world, with more recent fossils appearing in Eurasia and Africa. These fossils exhibit distinct adaptations to those differing environments. Later, about 1.5 million years ago, much of the horse diversity, including all North American horse species, disappeared. Only a single genus or group of species remains on Earth called Equus. It is tempting to imagine a straight path from modern horses extending back through their 55 million year evolutionary history. But that's just not how evolution works. Numerous branches of horses have split off over evolutionary time, flourished for millions of years, and only recently gone extinct. What we see living today is just a single branch of a greatly branched evolutionary tree. So the fossil record provides another valuable piece of evidence for evolution in the form of fossils with transitional features. These fossils demonstrate a link between groups of species thought to have a shared common ancestor. One such fossil is Tiktaalik, first found in northern Canada and estimated to be 375 million years old. Tiktaalik fossils seem to represent a transitional phase between lobe-finned fishes and terrestrial vertebrates. These creatures, like lobe-finned fishes, had gills, scales, and fins, but they also had arm-like joints in their fins and could support their bodies with their limbs in much the same way that a salamander can. And that brings us to our take-home message for 10.15. By analyzing fossil remains, paleontologists can reconstruct what organisms looked like a long time ago. They can also learn how organisms were related to each other and understand how groups of organisms evolved over time. So, when it comes to species distribution, history matters. The species that arrive in a geographic region first, usually coming from nearby, make up numerous different lifestyles in numerous different habitats, and the populations adapt to and evolve in each environment. Biogeography, the study of the distribution patterns of living organisms around the world, was observed by Darwin and many subsequent researchers and provides strong evidence that evolutionary forces are responsible for these patterns. For example, species often more closely resemble other species that live less than 100 miles away, but in radically different habitats than they resemble species living thousands of miles away in nearly identical habitats. In Hawaii, for example, nearly every bird is some sort of modified honey creeper, from seed-eating honey creepers to curved-billed nectar-feeding honey creepers. Large, isolated habitats also have interesting biogeographic patterns. Australia and Madagascar are filled with unique organisms that are clearly not closely related to organisms elsewhere. In Australia, for example, marsupial species, rather than placental mammals, fill all of the usual roles. There are marsupial wolves, marsupial mice, marsupial squirrels, and marsupial anteaters. So, the marsupials of Australia physically resemble their placental counterparts, but molecular analysis shows they are more closely related to one another, sharing a common marsupial ancestor. Their relatedness is also revealed by similarities in their reproduction. Females give birth to offspring at a relatively early stage of development, and the offspring finish their development in a pouch. The presence of marsupials in Australia does not simply mean that marsupials are better adapted than placentals to Australian habitats. When placental organisms are transplanted to Australia, they do just fine, often thriving to the point of endangering the native species. Instead, it seems that the terrestrial placental mammals in Australia disappeared about 55 million years ago for reasons that are not clear, giving the marsupials the chance to flourish and diversify. Biogeographic patterns, such as those seen in honey creepers and in the marsupials of Australia, illustrate that evolution doesn't necessarily lead to the same quote-unquote solutions for each set of environmental conditions. Rather, the traits and populations that happen to be in a particular location gradually change, and those species become better adapted to the habitats they occupy. And that brings us to our take-home message for section 10.16. Observing geographic patterns of species distributions, noting similarities and differences among species living close together but in different habitats, and among species living in similar habitats but located far from one another, helps us to understand the evolutionary histories of populations. So, if you observe any vertebrate embryo, humans, turtles, chickens, or sharks, while it's developing, you will see that it passes through a stage in which it has little gill pouches on the sides of its neck. It also passes through a stage in which it has a long bony tail. The gill pouches disappear before birth in all but the fishes. Similarly, we don't find humans with tails. Why do these features exist during an embryo's development? Such shared embryological stages indicate that the organisms share a common ancestor from which all have been modified. So, among adult animals, several features of anatomy reveal the ghost of evolution in action. Many related organisms show similarities that can be explained only through evolutionary relatedness. The forelimbs of mammals such as bats, porpoises, horses, and humans are used for a variety of different functions. If each had been designed for the uses necessary to that species, flying, swimming, running, grasping, we would expect dramatically different designs, and yet all of these species possess the same bones, modified extensively, revealing that they share a common ancestor. These features are called homologous structures. Then, at the extreme, homologous structures sometimes come to have little or no function at all. Such evolutionary leftovers, called vestigial structures, exist because they had value in an ancestor species. Some vestigial structures in mammals include the molars that continued to grow in vampire bats, even though these bats have a completely liquid diet. Eye sockets with no eyes in some populations of cave-dwelling fishes, and in whales, pelvic bones that are attached to nothing, but in nearly all other mammals serve as important attachment points for leg bones. Even humans have vestigial traits, such as the fused vertebrae that form your coccyx or your tailbone. This is a remnant of the tail found in most mammals. So, not all organisms with similar-looking adaptations share recent ancestors. Consider, for example, flying mammals, bats, and flying insects, locusts. Likewise, dolphins and penguins live in similar habitats and have flippers that help them swim. In both examples, the analogous structures developed from different original structures. Natural selection, in a process called convergent evolution, may act on different starting materials, such as a flipper or a forelimb, and modify them until they serve a similar purpose, much as we saw in the marsupial and placental mammals. And that brings us to our take-home message for section 10.17, similarities in the anatomy and development of different groups of organisms and their physical appearances can reveal common evolutionary origins. Moving on, all living organisms share the same genetic code. From microscopic bacteria to flowering plants to insects and primates, the molecular instructions for building organisms and transmitting heredity information consists of 4 simple bases arranged in an almost unlimited variety of sequences. Related individuals within a species share a greater proportion of their DNA than do unrelated individuals. This is not unexpected. After all, you and your siblings got all of your DNA from the same 2 parents, while you and your cousins got half of your DNA from the same 2 grandparents. The more distantly you and other individuals are related, the more your DNA differs. We can measure the similarity between the DNA of 2 species by comparing their DNA sequences for individual genes. For example, let's look at a gene that codes for a polypeptide used to build the protein hemoglobin. In vertebrate animals, hemoglobin is found inside red blood cells where it functions to carry oxygen throughout the body. It is made up of 2 types of chains of amino acids, the alpha chain and the beta chain. In humans, the beta chain has 146 amino acids. In rhesus monkeys, the beta chain is nearly identical in sequence. Of the 146 amino acids, 138 are the same as those found in human hemoglobin, and only 8 are different. In dogs, the sequence is still somewhat similar, with 114 of the same amino acids. When we look at non-mammals such as birds, we find about 101 amino acid similarities with humans. And between humans and lamprey eels, still vertebrates, but with more than 500 million years since our last common ancestor, there are 21 amino acid similarities. So the differences in the amino acid sequence of the beta hemoglobin chain, and thus in the alleles that code for this structure, indicate that humans have more recently shared a common ancestor with rhesus monkeys than with dogs. Similarly, they suggest that we have more recently shared an ancestor with dogs than with birds or lampreys. These findings are just as we would expect, based on estimates of evolutionary relatedness made from comparative anatomy and embryology, as well as from the fossil record. It is as if a molecular clock is ticking. The longer two species have been evolving separately, the greater the number of changes in amino acid sequences, or ticks of the clock. And that brings us to our take-home message for section 10.18. All living organisms share the same genetic code. The degree of similarity in the DNA of different species can reveal how closely related they are and the amount of time that has passed since their last common ancestor. So we can observe and measure evolution as it is happening by choosing the right species, preferably organisms with short lifespans, and designing careful experiments. In one clever study, researchers looked at populations of grass plants on golf courses, a habitat where lawnmower blades represent a significant source of mortality. All the grass on the golf courses was of the same species, but on the putting greens, it was cut very frequently. On the fairways, it was cut only occasionally, and in the rough, it was almost never cut at all. Over just a few years, significant changes took place in these different grass populations. The grass plants on the putting greens rapidly developed to reproductive age and produced many seeds. For the plants in these populations, life was short and reproduction came quickly. Plants on the fairways had slightly slower development and a reduced seed output, while those in the rough had the slowest development and the lowest seed output of all. When plants from each of the three habitats were collected and grown together in greenhouses under identical conditions without mowing, the dramatic differences in growth, development, and lifespan remained. The frequencies of alleles that control the traits of lifespan and reproductive output in the three populations had changed. In other words, evolution had occurred. So one particularly disturbing line of evidence for the occurrence of evolution in nature comes from the evolution of antibiotic-resistant strains of bacteria that cause illness in humans. In the 1940s, when penicillin was first used as a treatment for bacterial infections, it was uniformly effective in killing Staphylococcus aureus. However, some strains of Staphylococcus aureus contain alleles that make them resistant to penicillin. Because penicillin has become such a pervasive toxin in the environment of Staphylococcus, natural selection has led to an increase in the frequency of alleles that make these strains resistant to the drug. Today, humans are increasingly at risk for becoming infected with staph and getting diseases such as pneumonia and meningitis. Currently, more than 90% of the isolated staph strains are resistant to penicillin. In the 1960s, the antibiotics methicillin and oxacillin had nearly complete effectiveness against staph. But now, nearly a third of staph infections are resistant to these antibiotics as well. The meaning of such unintentional natural selection experiments is clear and consistent. Evolution is occurring all around us. Reflecting on both the process and the products of evolution and natural selection, in the final paragraph of The Origin of Species, Darwin eloquently wrote, "There is grandeur in this view of life, and that, From so simple a beginning, endless forms most beauty and most wonderful have been and are being evolved. And that brings us to our take-home message for section 10.19. Replicated, controlled laboratory selection experiments and long-term field studies of natural populations allow us to watch and measure evolution as it occurs.